we ended on Friday, I want to jump back here and make an important clarification. So this was really looking at the regulation of water excretion as GFR changes. So we know GFR is auto-regulated. It's kept at a very tight, it's kept in a very tight window at 125 milliliters per minute, as long as the blood pressure or the mean arterial pressure is between 80 and 180. So those three mechanisms that I mentioned are not pertaining to a GFR change. They're pertaining to keeping GFR regulated. And I really want to make that distinction here so that we're not confused. So the three mechanisms I talked about, tubular glomerular feedback, the mesangial cells, and myogenic regulation, are what keep GFR auto-regulated. These are what keep GFR at 125 milliliters per minute. If we move to, if our blood pressure rather drops less than 180, GFR will drop. We will no longer keep GFR at 125. It will drop. As a result, the amount of filtered water will drop, the amount of excreted water will drop as well. Likewise, if blood pressure goes above 180, GFR will increase, the amount of filtered water will increase, the amount of filtered or excreted water will increase as well. And these two events, right, raising or dropping the GFR, are brought about as a means of extrinsic regulation. So these are where we talked about the nervous system, neurons, and hormones getting involved to either drop the GFR if our pressure goes less than 80, or raise the GFR if pressure goes above 180. Okay, so I really want to make that clarification here. The next thing I want to do is go over the checkpoint question. So we had a few checkpoint questions we skipped over. I think we had two different groups of checkpoint questions. Let's see. Okay. So this was the first set of checkpoint questions here. And hopefully you would have already crafted a response to these, and we can kind of go over what an ideal answer would look like, and you can compare what you wrote to what I discussed now. So the first question is, where does most water reabsorption occur in the renal tubules? This, plain and simple, we know is the proximal convoluted tubule. We've got free water reabsorption happening here. One, it's unregulated, so it's happening irrespective of any external stimuli. Two, it's happening as a result of solute reabsorption. So the movement of solutes across the proximal convoluted tubule is what is facilitating the gradient for the movement of water. And that happens in the PCT. That's where 70% of the water is reabsorbed. The second part of that question, where does the regulation of water reabsorption in the renal tubules occur? So where does the regulated water reabsorption occur, right? So in the PCT, it's unregulated. In the distal tubule and in the collecting duct, it is regulated. And when we say regulated, we mean that it's happening as a response to hormone secretion, specifically ADH or vasopressin. And so if we got more ADH, we're going to reabsorb more water. If we got less ADH, we're going to reabsorb less water. So this is what we mean by being regulated, the water reabsorption here being regulated. And this regulated reabsorption takes place in the distal tubule and in the collecting duct. The second question, compare the movements of solute and water across the epithelial cells lining the descending limb of the loop of Henle and those lining the ascending limb of the loop of Henle, and then explain how this differential movement allows the establishment of an osmotic gradient in the renal medulla. So this is what we talked about last time. The disparity between the permeability of the ascending limb and the permeability of the descending limb. And if we jump back to this slide, we know, we can see here that the ascending limb is impermeable to water. The descending limb is impermeable to solutes. So along the ascending limb, we're only moving sodium chloride and potassium, no water. Along the descending limb, we're only moving water, no solutes. And this is how we establish that gradient. We call it the medullary osmotic gradient, okay? And if you look at this sequence of events that we kind of went through step by step, this is what sets up that gradient. The fact that solutes moves across the ascending limb, water moves across the descending limb, and then we push that tubular fluid a little bit more down as more fluid comes in to the descending limb, and then we start that process over again, and then we push that fluid down, we start that process again, push that fluid down. That eventually ends up with a scenario where we've got this gradient. The interstitial fluid that is closer to the cortex has a lower osmolarity. The interstitial fluid that is closer to the medulla, that's deeper in the medulla, has a higher osmolarity, and that is the medullary osmotic gradient, right? There's a change in the osmolarity as we go from cortex to medulla. And so that is set up by the fact that we have those differing permeabilities down the descending and the ascending limb, respectively. Okay. Let's go back here. Where's that slide? Okay. And then the third question is, can pure water be excreted as urine? And the answer here is no. Okay. And if we understand, one, what pure water is, and two, the production of urine, we know that pure water can never be excreted as urine. Okay. First of all, we filter our plasma across the glomerulus. And so we end up with this filtrate that has all of these solutes from the plasma, some of which are reabsorbed, some of which are secreted, but they must have a minimal osmolarity of 100 milliosmoles. And so meaning that at the most dilute, even in the most dilute scenario, the most dilute instance, or the most dilute urine, there is 100 milliosmoles of particles dissolved. Water has an osmolarity of 0, meaning that there's absolutely no solutes in that concentration in that fluid. And so we cannot get an osmolarity that is less than 100 milliosmoles, meaning that we cannot excrete pure water. There must be some solute present, even in the most dilute state of urine, okay? So the short answer to that question was no. All right, so I'll pause here, see if there are any questions that pop up in response to those checkpoint questions. I don't see anything, so let's jump to the other set of questions. I believe they were at the end. Okay, so there were 2 checkpoint questions here at the end. One, name the 2 stimuli for ADH released from the posterior pituitary. And let's talk about what ADH does first. So ADH is the hormone released from the posterior pituitary. It's going to go to our principal cells of the distal tubule and the collecting duct. It's going to stimulate those 2 cells to insert aquaporin channels into, here we go, into the basal, into the apical membrane. And that is going to facilitate the reabsorption of water, right? So that's what ADH is doing. And so the stimuli for ADH is going to be decreased plasma concentration, decreased blood pressure, decreased blood volume. The first one we talked about was increased plasma osmolarity, meaning that we have more salty or more concentrated plasma. And then the second stimulus was decreased baroreceptor stretch. I think that was on this slide. So if we think about it, if we decrease our blood volume, decrease our blood pressure, we're gonna have a more concentrated plasma. That means our plasma concentration, our plasma osmolarity will be really high. That is one stimulus for ADH production or ADH secretion. The second stimulus was the baroreceptor activity. And if we remember the location of the baroreceptors, they're going to be on our blood vessels, right, where we have the carotid sinus, and they're going to be detecting the level of stretch in that vessel, which is another way of saying the level of pressure in those vessels. And if we decrease blood pressure, we're going to decrease baroreceptor stretch. That is a second stimulus for ADH secretion in order to reabsorb more water to regulate that blood pressure, to get the blood pressure back up. Okay, so the two stimuli for ADH secretion are increased plasma osmolarity and decrease baroreceptor stretch. The second question here, describe the effects of ADH on principal cells. Okay, we kind of just went through that, right? ADH is going to signal a whole sequence of events inside the cells, binding to that metabotropic receptor, increasing cyclic AMP. That is going to insert these aquaporin 2 channels into the apical surface, brings water into the cell, and then we know water can leave the cell out the basolateral surface via the aquaporin 3 channels. Okay, so ADH essentially allows the principal cells to reabsorb more water back into the plasma. Okay, and I'll check if there are any questions on those. Otherwise, we'll look at the other questions I wanted to go over. Okay, I don't see anything popping up here, so let's look at this checkpoint, this weekly quiz question. So this question had a bit of disparity in terms of the responses. We had kind of a bit of mixed responses for this weekly quiz question. And the question was, the clearance of what substance, a natural byproduct of muscle metabolism, provides a non-invasive estimate of GFR, glomerular filtration rate? And the answer was creatinine. Okay, so there were two important concepts to be able to get this question right. One was understanding that creatinine is a natural byproduct of muscle metabolism, so it's a breakdown product when we metabolize muscle, but also it is a non-invasive estimate of GFR. So when we talk about estimating GFR, we want to look for a substance that's going to be neither secreted nor reabsorbed. It's going to move through the tubular structures and not be secreted or reabsorbed in excess, okay? And so urea is not going to be that. Urea is very much reabsorbed. Urea is involved in facilitating the reabsorption of water. And so we talked about those urea transporters that facilitates the reabsorption of water. So urea has a clearance rate that is very, very low, meaning that it is extensively reabsorbed. I think the urea clearance rate is about 1 milliliter per minute. And when we're looking for a substance that is going to estimate GFR, we want to look for a substance that has a clearance rate that is close to GFR. That is going to be creatinine, which has a clearance rate of 140, so kind of close to what GFR looks like, or inulin. Inulin is another substance that has a clearance rate of 125 milliliters per minute, which is exactly matching what GFR is, right? 125 milliliters per minute. The reason inulin is wrong here is that inulin is, one, not a byproduct of muscle metabolism. Two, it's not a non-invasive method of estimating GFR. It is, in fact, very invasive. And I guess you would have had to have an understanding of how we use inulin in order to get this question right, outside of knowing that inulin is not a product of muscle metabolism. Inulin is found in roots, I think like dandelion. It's found in plants. It's actually a polysaccharide. It's a carbohydrate. And we actually infuse it into the vein. So we have to put it exogenously into our body and then look at how it is cleared through the urine. So this is a very invasive mechanism of estimating GFR, right? We don't have inulin in our bodies naturally. And so in order to use inulin to estimate GFR, we're gonna have to shoot it into our veins or administer it exogenously, and then look at how it clears to the urine. So two important concepts to get this question right was knowing that creatinine is the only product here that is a byproduct of muscle metabolism. And two, creatinine is the only natural substance that has a clearance rate that is close to GFR, okay? I hope that made sense. The second question that I was asked to kind of review here was this question. So the osmotic gradient in the medullary region of the kidneys is established primarily by which of the following? And the answer here was sodium, potassium, chloride co-transporters on the ascending limb of the loop of Henle. So this question is basically asking, how do we establish the medullary osmotic gradient? And if we go back to this slide, where we talked about the establishment of that gradient, the first event, the first thing that has to happen to get this gradient set up is that sodium, chloride, and potassium must be actively transported across the ascending limb of the loop of Henle. Okay, that is the first part of setting up this gradient. That is the first part of establishing this gradient. The movement of sodium, chloride, and potassium actively across the ascending limb of the loop of Henle. Okay, so I hope that made sense. The other options here, so the efflux of water is basically happening secondary to the movement of those solids. So this is not going to be correct. And then this is not correct because this is not happening on the descending limb.